Hey guys, this is the lecture 3 finishing up this. I'm sorry I made a mistake when I looked at that video topoisomerase. Once again, these are the 6 enzymes you need to know: helicase primase, topoisomerase, DNA polymerase 3, DNA polymerase 1, and DNA ligase. You should know what their functions are, and we'll look at those real quickly. Okay, so DNA replication occurs at replication forks, which are inside something we call the replication bubble. Okay, and so what we see here is we see helicase, which is unwinding the DNA. Now, what's not being shown is topoisomerase, which would be untorquing the highly supercoiled DNA and making it less coiled, so helicase can come in and start to split those two strands. The SSBs are the single-stranded binding proteins. They're going to stabilize that replication fork, keep it open. And remember, we have the two strands. We have a leading strand, which is going to be copied continuously because it's actually following the helicase and is being made in the proper orientation, 5' to 3'. Remember, we can only add nucleotides to the 3' end. And since this is the leading strand, the has the 3' here, which means that the new strand will be 5'. Remember, we can only make in 5' to 3', so it's making it continuously. And the lagging strand, however, this is the 5' end, which means this is the 3' end. We can't make it this way. It has to be made from 5' to 3'. So as helicase moves down, it's going to keep on exposing new uncopied DNA, new template, so to speak, which has to go through the whole process where you have to put a primer on. Primase is going to come in, put an RNA primer. DNA polymerase is going to bind to that RNA primer, and it's going to add nucleotides in the correct direction, and then it will disengage once it hits another RNA primer. Where's that other RNA primer coming from? Well, as helicase moves down, it exposes more DNA template. We have to put another primer on, so on and so on. So on the lagging strand, what you end up with is a whole series of short fragments that we call Okazaki fragments, and all of these primers, and they're not showing it, but there's a primer here on the leading strand also, have to be replaced by DNA. Well, the enzyme that does that is DNA polymerase 1. So polymerase 1 comes in, replaces the RNA primer. You see there's a little space right there because it can't get all of it, so to speak. And then DNA ligase will come in. DNA ligase will come in and hook up that sugar phosphate backbone. Remember, DNA is a ladder. Let's go back a little bit right to here. DNA is like a ladder-like structure. That's what we call it, the ladder of life. The sides of the ladder are made up of sugar, phosphate, sugar, phosphate, sugar, phosphate. So we call it the sugar phosphate backbone. Whereas the rungs of the ladder, what you would put your feet on or your hands on as you're climbing the ladder, are made up of base pairs of nitrogenous bases. Do remember, cytosine and guanine have 3 hydrogen bonds. Thymine and adenine only have 2 hydrogen bonds between them. Cytosine and thymine are pyrimidines. Guanine and adenine are purines. Okay, remember, big structure, short word, purine, little structure, big word, pyrimidine. Okay, but the CG combinations have 3 bonds. The TA combinations only have 2 bonds. And as you can imagine, these are slightly harder to break apart than these are. Okay, so just kind of looking a little bit back at structure once again. So let's look at a short little video of DNA replication. Okay, oops, excuse me. Go back to here. I gotta see if I can hit that. DNA replication. Here we go. All right, so I'm going to kind of narrate this as we move along because I don't think you'll be able to hear the sound, although you can always click on that and watch it yourself.
The replication of DNA begins at a sequence of nucleotides called the origin of replication.
So there's the origin of
Helicase
replication.
unwinds the double-stranded DNA helix, and
Helicase is
single-strand
going to land
binding
at the origin of
proteins
replication.
react with
It's
the
going to
single-stranded
unwind the
regions
DNA.
of the DNA and
Topoisomerase,
stabilize
once
it.
again, is not being shown. Single-stranded binding
DNA polymerase
proteins are
3
going to
is the
stabilize
major
that.
enzyme involved
DNA
in DNA
polymerase, again, is
replication.
the major enzyme
DNA
coming in.
polymerase 3
It is
can
going
only
to
add a
add
nucleotide to the 3' end
nucleosides
of a
to the
pre-existing
3'
chain of
end.
nucleotides,
And remember,
it
the
cannot
nucleosides
initiate a
contain the
nucleotide
energy
chain.
to do that anabolic
Therefore, an
reaction, which
RNA
is
polymerase, called a
endergonic.
primase, constructs
Okay,
an
so,
RNA primer, a sequence of about
primase,
10
remember, is our first
nucleotides
enzyme.
complementary
Actually,
to the parent
it's going to add
DNA.
the RNA primer.
DNA
This model is
polymerase
a little
3 can then
incorrect.
add
I'm going to stop it for a sec. This model is a little incorrect right here because basically in a replication bubble, the replication would be going in both directions. Here they're only showing it in one direction. So this might be an example that you would see in a prokaryote, but not in a eukaryote. Okay, so in a bacteria, this is the way you would see it happening, but in a eukaryote, you would actually have DNA replication going to the right, and you would have DNA replication going to the left.
Nucleotides
so once again, DNA polymerase has to bind to RNA primer.
DNA.
It is now going to add nucleotides in the
because
5'
the two
to
parent
3'
strands of
direction
DNA
of
are
the new strand, not the old
they are
strand,
oriented
the new
in
strand.
opposite directions and
Okay.
must therefore be elongated by different mechanisms.
Remember, the strands are
leading strand
anti-parallel, so
elongates
they're
toward the
oriented in different
replication fork
directions.
by adding
The leading strand
nucleotides
is going
continuously
to be
to its growing
synthesized
3' end.
continuously,
In
following
contrast,
the
the lagging
helicase,
strand,
whereas the
which
lagging
elongates
strand
away from the
is
replication
going to be
fork,
synthesized discontinuously in a series of short
short
Okazaki fragments. Again, we're
called
exposing
Okazaki
new
fragments.
bare
When the DNA polymerase 3 reaches
template,
the RNA
we have to
primer
put
on the lagging
the
strand,
primer
it
down.
is replaced
Polymerase
by
comes in.
DNA
We got more
polymerase
template.
1, which removes the RNA and
We got to put
replaces
another
it with
primer
DNA.
in.
DNA
Okay, ligase
ligase
is
then
hooking
attaches
up that
and
sugar
forms
phosphate
phosphodiester
backbone.
bonds. The DNA is further unwound, new
There goes another
primers are
primer.
made,
DNA
and
polymerase
DNA
once again
polymerase 3
going
jumps
ahead and
ahead
doing it.
to begin synthesizing
Okay.
another
Now this is a
Okazaki
simplified
fragment.
version. It's much more complicated than that, and we'll see that in a second here. Okay.
For
This is the
simplicity,
way it actually
DNA
looks.
polymerase 3 has been
there's
depicted
two
as separate units,
It's called a
one
dimer,
acting on the leading strand and the other acting on the lagging strand.
Okay.
The current
And we
view
see
of
on the
DNA
lagging
polymerase
strand,
3 is that the two
the
subunits function
the DNA
together with the
template is
DNA on the
actually
lagging strand,
looped and
folding
going
to allow the
through
dimeric DNA
polymerase backwards.
molecule
So
to
we
replicate
see
both
this
strands
loop
of the
right
parental
here,
DNA
okay,
duplex
which we don't see
simultaneously.
in the leading strand. But we see
Proteins
this little
other than
loop coming
DNA
through.
polymerase 3 are
That
not
has
shown.
to happen. And we're going to look at another video of that in a second. So let's go back over to here and continue onwards. Okay, so as I said earlier, in eukaryotes, this does happen in a bubble where we see DNA replication occurring in both directions at the same time. Do notice, though, however, as you change directions, then the leading and lagging strands also kind of change. So going to the right, this is the 5' end here. That means this is 3' right here. That means that would make this 5'. Okay, so going this way, this will be the leading strand. However, and this will be the lagging strand, which will be done discontinuously. However, with the DNA helicase right here moving in this direction, this is 5'. That means this is going to be 3' up here, and this will be 5' to 3'. So this will now be the leading strand, and this will be the lagging strand, and they will meet somewhere in the middle. And we're going to have to go through with DNA polymerase 1, removing all the primers, ligase coming in, and attaching those sugar phosphate backbones. So what's the benefit of this? It reduces the amount of time in a prokaryote, which has a very small amount of DNA. Okay, this DNA replication, there's only one origin of replication in a prokaryotic cell as it undergoes binary fission. Okay, this can happen in as little as about 10 to 15 minutes. It can replicate all of its DNA. All right, however, in a eukaryote, which has 100 to 1,000 times as much DNA, multiple chromosomes, you have to have a origin of replication on each chromosome. And then on each chromosome, there are multiple origins of replication in order for DNA to replicate relatively quickly. And in an organism such as our cells, we can replicate our entire DNA, all 3 billion bases. There's actually 3 billion base pairs, so we're going to have to, hook up 6 billion nucleotides to make 2 new copies of DNA. Okay, to make those 2 new copies of DNA, add those 6 billion nucleotides. That only takes about 8 hours. Remember, for you to count to 1 billion with one number per second, it would take you 37 years. The fact that this can be done in 8 hours, 3 billion base pairs, 6 billion nucleotides added to that DNA is extraordinary and so freaking cool. Okay, so here's just another a little breakdown of it. We're looking at the direction of the helicase unwinding is going to go from the left to the right. And remember, we can only add in the 5' to 3' direction. So we see this strand is 3'. That means this is going to be 5' to 3'. So this would be the leading strand. This strand down here, this is the 5' end, which means that this is going to be the 3' end. So we have to go 5' to 3' in this direction. So that's the lagging strand. As helicase moves on down, okay, the polymerase is going to be following it. Okay, and of course on the lagging strand, we're going to be producing those Okazaki fragments. So this is a little video on how DNA polymerase 3 works. So let's go ahead and watch this video real quickly. Let me bring it on down to here. Let's click that. Let's expand it on out.
During DNA replication,
so this is what it looks like. We got both strands coming in to helicase. This is the helicase molecule right here.
Copying occurs at a
these are the
localized
2
region called the
polymerase 3
fork,
molecules.
which is a Y-shaped structure.
All right, they're attached together, forming a dimer, 2 parts.
By a multi-enzyme
okay, so
complex.
helicase is spinning at about 10,000 RPMs. It is moving extremely fast. And if I remember, I think it's like, I can't remember if it's 600 nucleotides per minute or 600 nucleotides per second, but it's moving very, very, very quickly. So helicase is separating those strands right there. You can see that happening.
At 10,000 RPM,
moving at 10,000
in the
RPMs
case of bacterial systems.
in bacterial systems. I'm not sure how fast it moves in eukaryotes. All right, so here's our leading strand. Here's our lagging strand, the 5' strand.
Distinguished by the direction in which their component nucleotides join up.
So we're going to focus first on the leading strand.
The 3' DNA strand, also known as the leading strand,
we see that continual replication. There's my template going through polymerase, which is going to be adding nucleotides according to the template. And it's going to be done continuously.
Known as the lagging
However, on the lagging strand,
has the opposite
we're going to see that the DNA is going to come out
copying mechanism
and get looped out backwards.
As it emerges from the helicase, the lagging strand is organized into sections called Okazaki
See, there's
fragments.
The loop coming out backwards right there.
These are then presented to a second DNA polymerase enzyme in the preferred 5' to 3'
It's complicated, guys. I know you don't need to know all the details. The most important thing to recognize is that it's happening at a slower pace because of the structure of DNA. This is the mechanism that has evolved over billions of years to replicate. Probably not over maybe a billion, not even a billion years, probably maybe a billion years. Okay, very early on in life to replicate this DNA.
And in some systems, multiple Okazaki fragments may be present.
All right, so that is how that process works. Again, you don't need to know all the details of it. Let's get out of here. And let's go back to here. But I do want you to understand that the lagging strand is a much more complex process. It's a slower process. Remember, evolution doesn't make things brand new. It manipulates existing structures through the processes of mutation and natural selection. Okay, and you know, the reason why this is probably happening this way is a evolutionary a piece of baggage, so to speak. All right. There's our dimer part. Again, in eukaryotes, there are multiple points of origins of replication. So here's a DNA a chromosome. Here is one bubble. Here's a second bubble, third and fourth bubble. Replication is going in both directions, and as I said earlier, it takes about 8 hours to do this. Now, the other function that DNA polymerase does is it repairs mistakes. It's a proofreader, number one. It checks to see if a mistake has been made, and if a mistake is made, it will fix it. And it will also play a role in DNA that has been damaged by, let's say, mutagens such as UV radiation. Okay, it can come in and replace the DNA. Endonucleases, which we're going to learn about a little bit later, those are sometimes called DNA scissors. They will cut out the DNA in specific locations or remove it. DNA polymerase 3 will come in, replace that damaged DNA, and then the little nicks will be closed by, once again, DNA ligase. So what I do want to say is that DNA viruses, which there are quite a few, are relatively stable. They don't mutate much. However, viruses that use RNA as their genetic material, such as HIV, COVID-19, the SARS virus, the flu virus, all of those use RNA as a genetic material, and RNA polymerase does not have that proofreading ability. And simply because mistakes are made during the RNA replication process, mutations occur very frequently in RNA-based viruses, which makes it very difficult to actually create a vaccine against that RNA-based virus. And we're seeing that already with COVID. We were amazingly, the fact that we created a vaccine that is 95% effective in less than a year is a testament to the work done by thousands of scientists in the past to figure out all this stuff, thousands of scientists working now to do this, and the ability of science to share that information. China shared the sequence of the virus as soon as they had it sequenced, shared that information out. Other countries are sequencing the virus. This is how we figured out that we have a variant. We've had lots of variants. We're going to look at this in depth in this particular unit. We're also going to look at it in the evolution unit that we do, because it's just a fascinating story of what's going on here, and it's a great example of natural selection. Now, we have this variant that's now more highly contagious. It doesn't seem to be more deadly so far, but it has a paper was just published like 2 days ago that's proved that it is more transmissible than the current virus. And again, first showed up in England, but I guarantee you we do have it here. It was detected in Florida just last week. It was detected in California. It was detected in Colorado. And the U.S. does a lousy job of sequencing our viruses, whereas England does a great job. They sequence 10% of all the tests that they do of COVID positive patients. We do something like 0.03%, okay, because we don't believe in funding. We think it's all has to be done by individuals and stuff like that. So anyways, we are behind the 8 ball on this, and we are not doing a good job. And one of the reasons why we have one of the worst caseloads in the world in terms of COVID. We need to do better, guys, and we can do better, and it's going to be up to you to make sure that we do better. So I hope some of you guys go into sciences. I hope some of you go into politics in order to make the funds available. That's my soapbox moment for the day, so I'm going to stop. Anyways, when DNA replicates, we got a problem when we get to the end of the chromosome, okay? Because on the lagging strand, okay, there's going to be a primer that once it gets removed, DNA polymerase cannot bind to it because it's naked DNA. Okay, it's a little bit different. DNA polymerase 1 can't even bind to this, so we got this little overhanging piece that does not have a chunk of DNA there. Does not have its sister strand template. So when we undergo another round of replication, that chromosome is going to get shorter, and when it replicates again, it's going to get shorter and shorter and shorter and shorter. Okay, so how do we fix this problem? Well, we fix it with something called telomeres or telomeres. Okay, what are telomeres? They are expendable non-coding sequences at the end of all chromosomes. Okay, they are short sequences of bases repeated thousands of times. Now, this repeat sequence is different in different species. In humans, it happens to be TTA-GGG, and that's repeated thousands and thousands and thousands of times. There's an enzyme called telomerase that actually adds those telomere segments to the ends of chromosomes only in germ cells, okay, the end cells of the early stage embryo. After that, telomerase becomes inactive and it no longer does it. So each chromosome has only a certain number of these telomere repeats. And every time that chromosome undergoes division or replications, and in the process of cell division, mitosis or meiosis, that telomere becomes shorter. And we now know that this is a cause of aging because once that telomere gets to a certain length, that cell will undergo apoptosis. And we know that apoptosis is the primary cause of aging. So if we could figure out how to reactivate telomerase, we could potentially have an ageless cell, maybe an ageless being. Interesting. There's a tremendous amount of research going in on this, as you can imagine. But what are the ethics of this? This is a huge, huge ethical question and social dilemma. Who would get it? Who is going to benefit? How do you pay for it? Tremendous amounts of questions. We need people that are going to be looking at the ethics of this. So if you're interested in that, that's one of the things you like to look at. Bioethics is a great field to look at. So if you want to look at how this actually works, you can click on that link at the bottom of this. Okay. I'm not going to go through it here, but you can look at that link and see how it works. I just need you to know what a telomere is and basically what it does. It protects the ends of the DNA. And that is it.